Hello, everyone. Thank you for joining our webinar, The Good, the Bad, the Ugly, Identifying Foreign Materials and Raw Materials and Finished Products, hosted by Mike Dziewatkoski of Eurofins SF Analytical. I'm Genevieve Randall, and I'll be moderating this webinar. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded, and slides and a recording will be available for you in 2 business days. A 15-minute Q&A session will follow the presentation to answer any viewer-submitted questions from you. During this webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit Enter on your keyboard to submit your question. Now I'll give an overview of Eurofins. Eurofins is driven by our mission to contribute to global health by offering the highest quality testing, training, auditing, and consulting services. We strive to listen to our customers and not simply meet but exceed their expectations. Our footprint is global, with 35,000 staff and 400 laboratories across 44 countries, and a portfolio of over 150,000 analytical methods. Eurofins provides a unique range of analytical testing services to the pharmaceutical, food, environmental, and consumer products industries and to governments. Now I will pass this webinar off to Mike. All right, Mike, you're free to begin.
All right, thanks, Genevieve. Hello, everyone, and I want to thank you all for joining us today. And what we're going to do is we're going to have a conversation about how raw material or foreign material identifications are performed. So when we're doing this work, we're talking about something that is in a product or a material or an ingredient that shouldn't normally be there. It's not expected to be there. So we're going to talk about some of the testing that is done to identify these materials. We're going to also talk about how the techniques we use are applied. And then we're going to go into some detail about actual case studies that we have encountered over the years. So to start off with, let's talk a little bit about some definitions and background. So, and we're going to be talking about how this relates to food safety. We're then going to be discussing the types of analyses that we commonly perform, the techniques we use, what type of data we receive, how we interpret the data, and then how we go about actually performing an identification. We're then going to be discussing what type of laboratories can do this work. So what are the requirements for a testing laboratory? And we're going to see there are a couple different types of testing laboratories, and one is really good at this type of work. Then we're going to be taking a look at how the results that you would receive from such a study are used, and then be taking a look at some case studies of actual real-world examples. So let's start off with some definitions and background. So what is a foreign material? Sometimes this is also called a foreign object. It's really anything in a raw material, a product, an ingredient, something you're manufacturing that doesn't belong. It's not expected to be there. It's not normally found in that product or that material. So it's called a foreign material or a foreign object. Sometimes it's referred to as extraneous matter. And the extraneous matter essentially is just an analogous term. The composition can be almost anything. It can be from an animal, could be a piece of a plant, it might be a piece of metal from part of the manufacturing equipment. It could be a shard of glass or a chunk of plastic that breaks off from a production line. Now these typically are all things we can easily see. Sometimes the material is invisible. We cannot readily see it. So it might be of a chemical nature. And it's totally clear, no color observed, and so it's truly something that we would have to investigate further to identify. Usually, this is of great concern related to food, but this really can occur for any type of a product. So we're going to keep this somewhat general today, and we're going to be not just talking about food, but a lot of different products that you might encounter. So how does this relate to safety and quality of the product? And we can ask a question, why do we do this testing? Our goal here is to identify the contaminant and to do that as early as possible. So here, we want to be able to perform this identification before we actually release a product into final production or for sale. This will ensure that what we're producing is of the highest quality possible. So the quality of the product hasn't been compromised. We also want to be able to determine root cause. So here we're determining what is the source of the contamination, why did it happen, and how can we prevent it. We're also going to do this work to prevent recalls. Since recalls are extremely expensive from the standpoint of not just the cost associated with pulling product, but also brand damage, brand reputation damage, we want to try to prevent a recall from happening. Now also, we'll find this being used, this type of work, for studying adulteration. And where adulteration comes into play is when a particular product is actually has something else introduced to it. So for example, honey being adulterated with high fructose corn sweetener. In this case, that's usually done to cheapen the product for an economic advantage. But it's good to be able to identify that to make sure, in this case, you're getting your money's worth in the product that you're purchasing. Sometimes the work is done in support of a legal claim. Here, it might be a situation where litigation is pending, and so the lab test laboratory is approached by a law firm in order to do testing on a product, and one way or another decide if there isn't a valid legal claim associated with a litigation. There could also be an insurance claim. Here, we're talking about a situation where perhaps a manufacturer has received product that is contaminated, and it might be several thousand tons of product, so of a huge cost. So who bears the cost of having that product destroyed? Is it the manufacturer, or can they file an insurance claim basis and actually have some recovery for damages? So these are all important questions as we answer why we're doing this. Now let's move on to the types of analysis that we actually perform. But before we do that, let's take a look at some of the most common recall products and why products are recalled. Here we have a table from FDA data from 2007, and it shows here the product that's recalled, the foreign material of concern, and then how much of the product was actually affected. And if you look at this table, what you'll see is that the products being recalled are quite varied in terms of the nature. So you could have olives or herring, finished products, baked products, raw food products, but you'll see that the materials that actually are found as contaminants could be metallic, could be plastic, could be glass. So these are all some of the more common ones that are encountered. You'll see that typically the amount of material impacted can be quite large, where you're talking about several thousand pounds or several thousand cases of a product. So here you can see the importance, since these are numerous occurrences, you can see the importance of how we're going to carry out this type of work. And so taking a look at the types of analyses used, we can think of different types in terms of those that are routine in nature, and routine analyses are what we commonly perform on samples for investigations. So for example, if you have a sample of a food product that has to be tested for pesticides, we know types of pesticides that need to be tested for. So we have a whole list. We look for just those pesticides. And the same thing is true with respect to heavy metals and residual solvents. We know what we're looking for when we perform routine testing. The situation changes when we actually doing specialized analysis. Here, for specialized analysis, we're looking for a true unknown. We don't know what the contaminant, what the foreign material is that is contaminating our product. We might have, by looking at the material, we might be able to draw some conclusions initially, but we have to go into some in-depth analysis using usually more than one technique in order to arrive at an answer. Now, as I mentioned early on, you can sometimes see what is the contaminant, but it's often invisible. So we're looking for contaminants that may affect how a product smells or how it tastes. So you open up a product and it just doesn't smell quite right. But it looks right. The color's right, the texture's right. So here we have to, again, use some very specialized testing to investigate why that is the case. So I think at some point, all of you have performed some sort of foreign material identification. I'm sure that at some point in time, you have been at a restaurant having a salad and you have found a hair in that salad. So what did you do aside from return it? What you did is you might have reached in, pulled out that hair, taken a look at it, and done what's called a macroscopic analysis. You've identified that, yeah, that's a hair, all right, and it's not supposed to be there. So I think most of you probably had some experience encountering that. Now let's take a closer look at how we're going to perform analyses in these cases. We can do an analysis using what is called a standardized method. And a standard method is one that has been developed by some organization where a group of people come together. They work as a committee to develop a method that they know works. It's been validated, it's proven to work, it's been tested multiple times, and has been shown to work. It can be applied to a variety of matrices. And so some of the organizations that do that type of work would be AOAC, AOCS, or EPA. Sometimes the work is also done by a particular company. So this might be a proprietary method to a company. But the method has gone through validation. It's been shown that it works. And so it's used for a standard method of analysis. Then there's another way of doing this testing, and that is with a customized approach. With a customized approach, you have to use unique ways of preparing the sample and then do the analysis in sometimes a very unique way, usually using more than one technique. So usually sample preparation involves maybe isolating a material, maybe adding a solvent and extracting, or maybe somehow performing a separation. It might be a crude filtration. But here, our goal is to isolate the material for further analysis. And as a simple example of doing an extraction, consider a material that is of a powder that is, let's say, contaminated with little dark colored flecks. And we might have an idea that maybe this is a metal, maybe it's magnetic. So literally we could take a magnet and hold it near the powder and indeed extract that material. In fact, we're doing an isolation, a separation, and an extraction all at once here. And so that's a very simple example, but that is actually a very effective way of doing sample preparation. Now, in terms of analysis, we're going to look at usually more than one technique, but we can also divide those techniques into different categories. So we can have general testing, we can do elemental analysis to look at the elemental composition of a material. We can perform molecular analysis where we're concerned with the identification or quantification of molecular species. We can use bioanalytical techniques that allow us to characterize proteins or bacteria. And then we can use imaging techniques to look at the morphology of a material. So now let's take a closer look, starting off with our general testing techniques. But before we get to that, we're going to do a poll question.
Okay, so for our polling question, does your company address contamination from foreign materials in its hazard analysis and critical control points plan? Yes or no? I'll leave the poll open for about a minute, and then I'll close it and share the results. Okay, I'm going to close the poll. And it looks like 91% do address contamination from foreign materials in their hazard analysis and critical control points plan. Okay. Mike, you can continue on with your presentation. Thank you, everyone.
All right. Thank you. Okay, now let's take a look at some of the techniques we actually use. And as I mentioned, starting into the poll question, we're going to take a look first at some of the general tests we perform. And many of you might be familiar with these general tests because they're relatively routine measurements. So measurements like pH and color, conductivity, and so on. Although these are routine and they're relatively simple, they can tell us a lot about what is happening to a sample. Now remember that I said that a foreign material contaminant could be invisible to us, at least invisible to us as we look at it with the unaided eye. So if you look at just considering pH, let's say, for example, your product typically starts out at has a pH of about neutral, roughly 7, maybe a little bit higher than 7. And after some time, the pH drops. And it drops to a very acidic condition. Let's say it goes to about pH 4 or something like that. That pH change may be indicating microbiological activity in that sample. So as investigating something that's going wrong with your sample contamination, we could look at, maybe you ought to do some microanalysis on the sample. Or maybe there are some side chemical reactions occurring from your product. The product isn't stable. And if you see that the pH is dropping, that might be an indication that you have a particular side reaction occurring, and if you know your product well, you might be able to actually identify what is happening. So pH is important, color change is important. Sometimes if you're looking at viscosity or even specific gravity, if a product has been diluted down, those are good indicators of that something else has been added to a product. And if a product has a chunk of glass in it, and it's a relatively large chunk of glass, that glass could be removed, and you could measure the refractive index of that glass, and it gives an indication sometimes of the type of glass it is as a first test. So these are actually tests that can guide us to further work, further study of the contamination. Now, what I need to point out here is that we have two general types of tests, or two types of tests that we can perform. Those that are destructive versus non-destructive. And so, for example, a pH test is non-destructive because we're not destroying the sample during that measurement. That sample is still available to us. As we start looking at some of the tests for elemental analysis and molecular analysis, those could be either destructive or non-destructive. So let's take a closer look now at techniques for elemental analysis. Some can be quite simple. Simple colorimetry, where we take a solution or a material, add a complexing agent to it, and it changes color. And many of you might be familiar with the lead tests that are sold in stores. The lead tests use a simple complexing agent, and if you see a pink color, you have lead present. The darker the pink color, the more lead you have present. We have other techniques that, for elemental analysis, that are a little bit more elaborate. So, like inductively coupled plasma optical emission spectrometry, or inductively coupled plasma mass spectrometry. Here, we're looking at the elements that are present, and we get spectral data. So shown here, this is an emission spectrum that we might get from a sample, and you can see a lot of different elements showing up here, like chromium, sodium, magnesium. And so we get a lot of information from a sample with regard to the elemental composition. We also might use a technique known as energy dispersive X-ray analysis, EDXA. This is a non-destructive technique, whereas the techniques here were destructive. So this technique gives us information about elemental composition, but we maintain the sample integrity. So we could use that sample later on for additional work. Sometimes we'll do work with ion selective electrodes that are specific for certain elements, and then sometimes ion chromatography, so looking at specific ions after they've been separated. With respect to molecular techniques, sometimes we use color tests. So for example, a simple color test for an aldehyde is called the tolerance test, where you actually mix it under right reaction conditions with silver ion in solution, and silver ion will plate. It will plate out to neutral silver, and silver metal will plate, and you'll see a shiny mirror appear. So that's a very simple color test. But we have more elaborate tests too for molecular analysis, and that would involve gas chromatography or liquid chromatography. And here we're getting some data with respect to a retention time for a compound and also a response. So this might be a response from a flame ionization detector, or maybe a response from a UV vis detector, so an ultraviolet visible detector, or we might even receive data from a mass spectrum of the compounds. But again, the reason we're doing this is to be able to quantify and identify the material that is contaminating our product. Other tests we use that actually come in quite handy are those that involve absorbance and transmittance. And we get a spectrum here of the amount of light absorbed or the amount of light transmitted through the sample with respect to different wavelengths. And One technique that is really great for the kind of work we do is infrared spectroscopy. So we're looking at the amount of light that is absorbed in the infrared region of the spectrum and relating that to a particular compound characteristic. So what this allows us to do is in the end be able to perform, again, unique identification using what is called fingerprinting from the compound. If we move into larger molecules, we have to use slightly different techniques. So here we're talking about big molecules like proteins, or maybe proteins that are allergens, and large living organisms like bacteria. Here we use molecular techniques like enzyme-linked immunosorbent assay, or some other immunoassays based on antibody techniques, or PCR, a polymerase chain reaction, to investigate nucleic acid content. And then sometimes we use antibodies in combination with PCR. But we're looking at proteins, relatively large molecules in this case, or proteins that might come from bacteria or molds or fungus, or perhaps nucleic acid sequences that are coming from those particular organisms. But we're trying to identify based on how they interact with antibodies, or or the composition of their DNA. Now, another powerful tool we use is microscopy. And there are two typical types we use, which is ordinary light microscopy, where we have usually magnifications on the order of maybe a couple hundred power, or we can use scanning electron microscopy, where magnifications are so high that we can actually observe particular features of materials that are on the order of a couple of micron. So very high magnification in this case. But what we have a chance to do is to look very carefully at the material and observe the morphology. So it tells us a lot about what is happening, or what has happened to the product from a contamination standpoint. Now I'm going to move into an area of what type of testing laboratories would perform this type of work. And this is important if you need to have this work done. How do you approach a particular laboratory to have the work done? So we can divide laboratories into two types. Those that are routine testing laboratories, and routine testing laboratories do a certain focus. They have a focus in certain areas. So for example, like food analysis or maybe environmental analysis. And they have a lot of experience in testing in that particular area. So it might be a lot of experience in food testing or pharmaceutical testing. So they're really good at it. And the reason they're really good at it is they do a lot of that testing. And so their focus is high throughput in this case. They want to be able to test a lot of samples on a daily basis. That's their business model. Now, in comparison, we can look at the non-routine testing lab. Here, this laboratory has more of a specialized focus. They're going to want to be able to solve very, very challenging problems. And they're going to use a lot of different techniques when they solve these challenging problems. And a foreign material identification is typically a challenging problem. So they're going to be able to use multiple techniques to arrive at an answer that is actually identifying the material. And typically what you'll find for these laboratories is that they have a lot of experience solving these types of problems, and they employ a lot of scientists. So people who make it their life's work to be able to really know how to use the techniques of analysis that we talked about a little earlier to actually be able to do these identifications. So they're really good at this type of work. Now, in terms of other characteristics and features of the test laboratory you engage, the test laboratory should be totally objective and independent, have no conflicts of interest whatsoever. It's incredibly important because especially if it is a situation where it is a legal claim or an insurance claim. They have to be able to have the capabilities of performing the analysis that you've asked them to perform. So if they're offering the service, you would expect them to be able to carry out that service. However, you might run into situations where you encounter not so really good result for a particular study. But you want to be able to characterize their capabilities, know what they can do, what they do well. They should be able to have the capacity to perform the work, have the instrumentation. They should have the ability to turn around or complete the project in a relatively short period of time, 2, 3, 4 weeks, something like that. But they also ought to be able to, if there is a delay, be able to communicate that with you, why that's happening. With respect to maintaining sample integrity, there has to be a strict chain of custody in place, and they should be able to have a way of storing samples in a secure location. And in the end, you could always ask for references for a laboratory from work that's been done for other clients in the past. It's entirely a valid thing to do. But up front, what you'd want to do is be able to ask a lot of questions and to come away feeling that you're satisfied in the capabilities of lab you have chosen. So what they're going to do is they're going to do work for you. They're going to do an analysis or multiple analyses and issue a report. But you have to figure out how to actually use that data. What's it going to tell you? How's it going to resolve your problem? So they're going to give to you, they're going to give their data. They may be a written report, a formal written report, more than just a summary of results. They might even interpret data for you, which they should. For this type of work, that's a requirement. That they've done, let's say, maybe 4 or 5 different tests. What each test say? What does it point to? And have we identified and maybe even quantified the foreign material? You should have access to scientists, people who are very skilled at performing these types of tests and who are very knowledgeable, and you should have the ability to ask them a lot of questions so that you are comfortable with the results you've received and that you can use those results to resolve the issue. And this, by way of that, we're going to determine the source or the cause of contamination. In addition, if this were to be a situation where of a legal nature, you might want to have the laboratory have a deposition done, affidavits generated, something like that, a testimonial. So they're going to, if they have the willingness to do, the ability to do that type of service for you, that's always a benefit in resolving a claim. So it's these types of results that we want to make sure that are being able to be used to resolve an issue that we're having. And now let's take another poll question.
Okay, so our next polling question is, in the past 2 years, how many times has your company conducted a foreign material identification study using an outside testing lab? 0 times, 1 time, 2 times, or more than 2 times? And I'll leave the poll open for a little while. Okay, I'm going to close the poll, and here are the results. Looks like most members of the audience have conducted a foreign material identification study with an outside testing lab, either not at all, or more than 2 times. All right, Mike, continue with the presentation. Thank you.
Great, thank you, everyone. Thank you for taking the poll. Now let's take a look at some actual case studies that we've encountered. And we're going to be looking at 5 situations here where we had a client that had a bottled juice product, and after a while, there was a gas formation problem. Another one where a cheese product, finished cheese product, actually had some particulate material found in it. A very interesting one where a bun was alleged to have been contaminated with rodent parts. And I think this one is very particularly interesting one because of the nature of it. A case where a pharmaceutical tablet was essentially was discolored a short time after being manufactured, and the client wasn't sure what was happening with their process. And then a bottled liquor product that, after it sat for a short period of time, was observed to have floating white material in it that obviously should not have been there. So let's start off with taking a look at a situation where we had a client come to us with the problem being that a product was hot packed juice, and just after a few weeks in the bottle, the product became cloudy, and there was a lot of gas formation. So literally, it foamed up after it being opened, and it wasn't supposed to happen. It should have been a shelf-stable product. So looking at this, what we decided to do was just based on what we know, the fact that it became cloudy and gas formation kind of hints that something contaminated. Maybe it was a fermentation going on, something growing in it. So we proceeded to perform yeast and mold testing, collect some of the liquid, and actually perform microscopy. And then we, based on these 2 early studies, we decided to move forward with some gene sequencing to perform an identification. So under microscopy, just ordinary light microscopy magnified a couple hundred times, this material was observed. And fortunately, we have some really great microbiologists on site, and the scientists actually looked at it and said, yeah, that's characteristic of actually a fungus. But just because you can see it under a microscope doesn't mean that, and it looks like it is, but we want to have some really definitive identification. So the gene sequencing was done, the actual microbiome of the material was performed, and it was turned out that it was indeed fungus contamination. Now, the way this occurred was that there was a product, what is actually just an additive to the juice, that sort of changes the characteristics of it to palatability, essentially, mouthfeel, and so on. And that particular additive was contaminated with a fungus. So it started to grow. And what happened was, it was really, the packaging process involved was modified that allowed to prevent this from happening. So this was a case where, again, using multiple different techniques, no chemistry was needed here, but a lot of microbiology, molecular techniques, and we were able to figure out what the root cause of the problem was. Next was a case of the cheese product, and here are actual images of that product where you could observe little black, reddish black, dark red, dark brown specks throughout the product, and it looked like pieces of metal. But what we found out was that through a variety of testing, that it wasn't actually metal. And here we had to perform non-destructive testing because we had to maintain and keep this material. We couldn't destroy it. So what we did was we did the SEM EDXA work, so scanning electron microscopy combined with the energy dispersive X-ray analysis, and we observed this type of granular material. Now, what's the great thing about SEM and EDXA combined is that you can pick off certain parts of material or interrogate small areas of a material and come up with the elemental composition shown here in this table. Well, if you're looking at the elemental composition here, you'll see that metallic elements content is very low, so iron content is extremely low. So it points to the fact it's probably not metallic in nature. But if you look at the carbon content, it's very high, as is the oxygen. So being that carbon and oxygen were quite high, metallic elements extremely low, or almost those that you would expect were non-existent, and based on the morphology, what the material looked like under magnification, we actually confirmed it to be some of the filter media that's used in packed filters. So there was just a disruption in the filter process, a breakthrough of the filter, and this ended up in their product. So that was an easy source to trace. Another case of the alleged rodent contamination in the bun. Here, this was a legal claim. So our client was being sued by a person who claimed to have found a piece of rodent in a bun, and well, essentially was claiming damages. So what we did was we used both microscopy and chemical analysis to compare the suspect bun to a reference bun. So the reference bun is the bun that the manufacturer would make all the time. It's supposed to be the same type of bun that the client claimed, or that the claim was filed against in terms of it being contaminated. So what we did was, for the chemical analysis, we compared protein and starch between the two buns, so a reference bun and the suspect bun. And what we found, though, was that there were very large differences between the two when we actually started looking at organic acids and preservatives. And really looking at using the liquid chromatography results, you can see that they were very, very different between the two materials. That's an indication that they were not the same. So what somebody did was they actually slipped in a bun, probably threw a piece of rodent in it, and tried to claim that it came from this manufacturer. So in the end, what it was is it came to a very favorable outcome for our client. So it was just a frivolous claim is what it was. Next case was of a pharmaceutical tablet that after being manufactured, there was discoloration observed. So the question becomes, what is happening here? Is it a contamination issue, or is there a formulation problem? And what we had, fortunately, was the suspect pill, the contaminated pill, and reference pill. So this is the normal pill right here. It's homogeneous in nature. It's all the same color across. This is a pill that is, we broke it apart, but this is the contamination. So what we were able to do is actually isolate the discolored spots from the normal spots and look at spatial variation across that material. And fortunately, we had the reference for comparison, which is critical here in this case, and using two different techniques, the FTIR, so in the infrared technique, and also SEM-EDXA. What it turns out was that the FTIR work was inconclusive. In this case, although it's a powerful technique, it provided us with little to no useful information. But SEM-EDXA did. What we're able to do, as I mentioned earlier, is we could isolate specific points on the tablet and perform an elemental analysis. And that's what was done here, points 4, 5, and 6, 4, 5, and 6, and compared to the control. And you'll see that the variation of one element in particular, and that's titanium, changed a lot across the spots. And it just so happens where the discoloration occurred is where the titanium concentration was the lowest. So it's either a reflectivity issue, meaning that the titanium was absent and so the pill didn't look white, or there could have been some photodegradation occurring in the tablet. But the problem is that it was a formulation issue. It was just it wasn't homogeneous during the pressing. So that was, again, in this case, a situation just happened. The manufacturer had to change the process slightly to account for that. And then finally, floating material in a liquor. In this case, this was a filtered liquor product. It was blended, and after being bottled a while, white flocculent material appeared within the bottle, and this occurred, this is a hint, it occurred after a water treatment process change. Okay, so that's some valuable information, a valuable clue. So what we did was we filtered, isolated that material, and under magnification, it appeared to be fibrous in nature. Okay, that's a clue. Then we did FTIR analysis on it, and in this case, it gave us some very conclusive information. So combining SCM and FTIR, SCM told us that it was of a fiber nature, and then FTIR told us that it was a melamine formaldehyde-based polymer. That was easy to trace back now to a water filter. And so that's what it was. It was actually contaminant from a water filter during processing. So a situation where combining a couple different techniques and paying attention to the clues that we observed, we were able to come up with an identification. And so now, to summarize, you can start off by saying that foreign material contamination is a problem really for any industry. So not just food. We observed it here in pharmaceutical, beverages. It can occur pretty much anywhere. It could also now be a situation where there might be an insurance claim involved or perhaps a legal claim involved. And as I hope you've observed that we have to use many different techniques, a wide array of techniques to be able to do this type of work. And you have to have the people and the knowledge to be able to apply those techniques to come to some conclusion in terms of identification. And then it's important to choose a lab that you're comfortable with and that can meet the needs of your study. So you have to choose very wisely here because the study outcome is important. The study outcome really depends upon the quality of the work that the laboratory does. And so with that, I'll thank you very much for your attendance today. I hope you found this webinar very useful, and we'll be happy to entertain any questions you may have.
Thank you, Mike, for your presentation. Now we'll move into a short Q&A session. Feel free to continue submitting questions during this session, and Mike will follow up with questions that he doesn't get to. Okay, first question. How would a law firm use these services?
Okay, thank you. So the way a law firm would use the services is that they're trying to build a case. They're trying to collect evidence. And what they have to do is they're trying to collect evidence in support of their case, obviously, in their position. And so what they would do is want to have the laboratory go through an in-depth analysis, so collect data that under very controlled conditions. So this is where samples have to be stored under very stable conditions, so they don't undergo degradation, and under very safe conditions. So not just anybody has access to the samples. They're very controlled. Then the analysis has to be done at a situation where it's all data is available for the law firm. So all raw data, all chromatograms, graphs, charts, you pretty much name it. They're going to have access to that because this data has to stand up to scrutiny. Okay, scrutiny from the opposition, obviously, for lawsuit. So that's how they're going to use the data, to use it in support of their case, but they have to make sure that it can stand up to scrutiny.
Okay. Next question. What type of documentation should accompany the sample in a legal case?
Could you repeat that one more time, please?
What type of documentation should accompany the sample in a legal case? Is that with respect? I'm having a little bit hard time here. We can follow up with this after the webinar individually.
Okay, that might be better. Okay.
Okay. Are there any USDA, FDA, or regulatory guidelines for foreign materials?
Well, in terms of the presence of a foreign material, a HACCP plan should address how you're going to deal with the presence of a foreign material. So what protocols do you have in place to be able to prevent such an occurrence? Now, in terms of identification, primarily all you're going to find is like a macro analytical guide. And there is some very basic guidance on how these studies are carried out. And that's really it. So, but when you're talking about macro analysis, you're talking about being able to see what is contaminating a product. Sometimes, as we've pointed out, these are such tiny, or sometimes it's invisible, the material that's contaminant. So those guidances aren't terribly applicable, but sometimes they can be of use. But typically what has to be done is that the laboratory performing the analysis has to have those guidelines in place, their own SOPs in place, and methods in place to be able to do this type of work.
Okay, thank you. We'll have another question. Any specific lab you can recommend for routine and specialized testing for foreign material?
Well, for routine testing, now let's say that if it's routine testing, you would want to know what you want to look for. So, for example, if you consider a residual solvent to be a foreign material contaminant, which it could be considered that, then what you do, you can go to a routine laboratory and ask them just to do a residual solvents test. So maybe the USP protocol for residual solvents. There, though, you typically want to know what you're looking for. So you want to have an idea of that. Now, with respect to non-routine work, you're going to, one of the better ways of doing it is to actually go to a web search and search for foreign material identification. There are also some publications on this work too, and that is that some of the trade journals, they will from time to time publish articles, and those who are writing the articles are associated with the test labs who are doing that type of work. So that's a good indication that they have a pretty good handle on the work they're doing. So I would suggest those different sources.
Okay. Next question. How often should routine testing be performed on raw materials from an approved supplier if the supplier supplies an allergen-free statement?
Well, the allergen-free statement, now that is a useful piece of information, but the question always falls upon the end user is it really allergen-free, or even just to say just contaminants in general, is it contaminant-free? So it typically falls upon the end user to actually show that it is free of a particular contaminant or meets a certain quality standard. So if a material is stated as complying with FCC, does it really? We do encounter situations where we have a COA for material and it's claimed to comply with USP, FCC, something like that, meet those requirements, but upon further testing, it doesn't. So the question becomes, how do you know when you should be testing, doing the confirmatory testing? And it really goes back to how much do you trust your supplier? And that's why that's a big topic right now from a regulatory standpoint. Do you trust your supplier? You have to be able to verify your supplier now that you're providing with a particular quality of material as you expect. So if in doubt, have the routine testing done to verify quality.
All right, next question. Okay. Is comprehensive data available on where an identification of foreign material occurs and at what frequencies?
Well, you can consult the FDA website for recalls in terms of what they're recalling for. So, for example, one of the most common reason a product is recalled is due to an allergen, that either allergen content that either wasn't claimed or allergen was found afterwards. So that's one of the most common reasons. So data like that is maintained by FDA. Now, in terms of, the question that was posed could be interpreted another way. How do you go about finding what the root cause of the contamination? That is typically a situation where, that's why pointing out during the presentation that being able to identify the material is crucial, and that leads to some indication where it could have come from. So, for example, if you find a piece of plastic in a material and it's of a particular nature, okay, let's say it's high density polyethylene, and it just so happens that your packaging material is not high density polyethylene, and the material did not come in a package that was high density polyethylene, but a production line might have a roller or a belt roller or something like that that is high density polyethylene, then there's some indication that it might have fractured off of that, fell into your product. So it's that type of information that allows you to backtrack. So it is crucial to get that identification performed, to know what the material is.
All right, we'll do another question. Mike, let me know if you want to follow up with this one after the webinar.
Okay.
Or not. When doing allergen verification on raw materials, do we test it for what allergen it has or what it doesn't have?
Well, so when you do allergen testing, you're looking for certain things. I know there are the big 8 allergens, okay? You're looking for those. So you're looking for those being present. Could there be something that is an allergen that could be present, something else that you didn't look for? Entirely possible. In some cases, sulfites can cause allergic reactions, and at very low levels. But you know, sometimes the products, if you don't expect sulfite to be present, it wouldn't be tested for it. So it's difficult to look for the unknown. That falls out of the routine realm and more into specialized testing. And in that case, then you would have to do very broad spectrum testing, but you'd have to have some idea of what you're looking for ideally. That would be the best approach. Ask the question, what do I want to look for? What could be in my product? And that's the best approach.
Okay, thanks. I think we have time for one or two questions left. For the rodent particles in the buns example, if the buns were the same, what would you do? And what testing would you do?
That is where we would actually do much more extensive testing, probably looking at maybe a carbohydrate profile and an amino acid profile, protein profile. What we would have to now start looking at instead of just what are considered to be some additives or compounds that are formed after the addition of yeast and the fermentation of yeast, we would actually have to look now more at the main structure of the material, which would be the flour, the gluten, perhaps gluten composition, the amount of gluten present, all of those things. So again, going from now what were low-level compounds, like the preservatives or the organic acids, to more the major components, look at those and see how they compare.
Okay. Let me look. Okay. Another question regarding FTIR analysis. How varied can a comparison sample be before declaring it to contain foreign materials? And this is our last question for the webinar.
Yeah, where we're looking in FTIR, primarily we're using the wavelength region that is at about roughly 800 to 2,000, maybe 2,500 wave numbers. And in this region, often it's referred to as the fingerprint region because it gives unique absorbance bands that point to certain functional groups. So the way you can see variation down there is that indeed you do have a very different material, and so you're not going to, so for the identification process, you're just not going to be able to use the functional group absorbance to identify the material. Perhaps the material is such a low concentration that the absorbance just isn't strong enough to give you a strong enough signal in that region. So that's one situation that you'd encounter where it wouldn't give good information for comparison. Typically, those types of techniques, they work better at higher concentrations, and so if you can isolate the material, and that's why that's so important, an extraction or an isolation, something like that, to isolate the material, effectively concentrate it, and then now you have enough to work with and to be able to perhaps get a strong enough absorbance to actually do the identification. So, but that's a great question, because if you have very, very complex mixtures, sometimes it's just not, you can't receive good information unless you actually do some isolation.
Okay, well, that wraps up our Q&A session. I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available through email within the next 2 business days. Thanks, Mike, for your expertise and your presentation. Thank you, everyone, for attending.
Thank you, everyone.